Hello, this is my first essay question assignment. And so the first thing we're going to look at today is what is homeostasis? I'll write that here. Homeostasis. When broken down into its Latin roots, homeo and stasis, homeo or homo meaning same, and stasis meaning not changing. Homeostasis literally means just staying the same. And how we use this in a biological context is the concept of homeostasis is that organisms are able to maintain their basic functions necessary for living. We are able to control our internal environment while our external environment may change, and it may change quite rapidly. Two main examples of this are our body temperature, because we're warm-blooded creatures, we maintain a relatively consistent internal body temperature, and also our blood pressure, which is necessary for cycling nutrients and transporting things throughout our body. And so let's have a look at a system as to how we might maintain homeostasis. So we start with a stimulus, and this can just be anything in our outside world that our body would need to respond to. It can be it being hot outside, it being cold outside, maybe we've been injured and we're losing a lot of blood. And so the stimulus is detected by a receptor. And now these can be like sensations, like on your skin, like you just touched a hot stove. And it can detect temperature, it can be our five senses, but something has to notice that the stimulus is there. And this receptor will use an afferent pathway. And the receptor will send a signal down this afferent pathway to the control center. And that afferent pathway is typically like our neuronal network. It can be our blood system, just any way that two different parts of the body can communicate with one another. And the receptor sends a signal through that afferent pathway to our control center. And we might typically think of that as the brain, but it doesn't necessarily have to be the brain. And so that control center will decide how the body needs to respond in order to maintain homeostasis, in order to make sure that this external environment does not wildly throw off the functions of the internal environment. And so the control center is going to use an efferent pathway, which is similar to an afferent pathway in that it's either like nerves or blood, just to get a signal through. And that efferent pathway will go to an effector. And this effector is going to be anything that will change something in how the body is doing in response to that stimulus. So just like, oh, it's hot outside, this is going to start making sweat, we're going to vasodilate in order to get some of that heat to our skin that it can radiate outwards. It's going to be your heart rate increasing in order to compensate for blood loss, to cycle as much blood as possible because there's not a lot of blood to go around. And so that effector will go back to the receptor and will want another kind of signal to see how it's doing. It's going to want feedback on how the system is now responding to the environment. Has the environment changed again, or did whatever the effector did fix whatever problem the receptor found? And this is what is called a feedback loop. The stimulus gives something to the receptor, which transfers down the afferent pathway to the control center, which decides what to do and sends that signal down the efferent pathway to the effector, which then cycles back to see what the receptor is doing. And so it just makes this loop. And in terms of feedback loops, there's two kinds we mainly see. We see negative feedback and we see positive feedback. And so negative feedback is what we see most often in our body. And the defining factor of negative feedback is that it brings the system back to its set point, back to its default. And so it's like, oh, it's hot outside, so a negative feedback loop would start implementing measures to cool down your body. It would want to bring your internal temperature back down to a more acceptable range. And with positive feedback, the defining feature of positive feedback is that it amplifies the effect of the stimulus. And it's comparable to kind of like a snowball effect. And one noteworthy example of this is during labor, is that contractions are controlled by a positive feedback loop. Because we have a contraction and that encourages the body to send more oxytocin, and the oxytocin will go out and will cause another contraction. It'll keep compounding on itself and they'll become stronger and they'll become more frequent until that baby is out. And so that's an example of a positive feedback loop. And so there is also something called a feed-forward mechanism, and that is when the system is anticipating a response. So it kind of gears itself up in anticipation. It's just like you're talking to your friend and there's this cool restaurant they want to go to that sounds really good, and your body is needing food. And so in anticipation of eating food soon, you're going to start salivating and producing those salivary digestive enzymes in anticipation of eating. And that is feedback systems. All right, looking at question 2 of our essay is we are looking at mediated transport. And so we can break this up into 2 major categories. And we have transport that does not require ATP, and that does require ATP. So looking at our no ATP first, the simplest form of this no ATP transport, this is often called passive transport, is we have passive diffusion. And so diffusion is simply the fact that molecules will travel from a concentration of, will travel down a gradient of high concentration to low concentration. Let's say if you have a handful of marbles and you put them on a sheet pan and let them roll around, after a while those marbles are going to be randomly scattered, but rather evenly distributed across that cookie sheet. There's not going to be more marbles on one side of the cookie sheet than the other if you're just going to let them roll around and distribute. And that's kind of the concept of passive diffusion. And so we see passive diffusion in cells with, we mainly see this with CO2 and O2, because the easiest things that travel across the membrane are small, non-polar molecules, just because the polarity of the molecules gets caught up in the lipid tails of the phospholipids, and they don't travel through very smoothly. And so small, non-polar, non-charged particles are going to be using this passive diffusion in order to move in and out of the cell. And there are other types of transport we can do without energy. We also have facilitated diffusion. And it essentially works the same as passive diffusion, except we now have a channel protein that allows for movement. So let's say this is our gate, and we have our cell here. Let's say we have 2 stars inside of the cell. These stars are large polar molecules like glucose or something of the like. They have a charge, they're large, they're spiky, it's hard for them to travel through the membrane. And so they are going to use this channel protein in order to enter into the cell and diffuse. Now the problem with this is that there's only one channel protein and there is a lot of stars outside the cell. So the problem with this is that these stars are going to have to line up single file in order to go through this channel, pretty much, because there's only one, and only one can go through at a time. It's like when you're entering a theme park and you have to walk through that gate with the 3 bars that spin as you walk through so they can count how many people come in. You have to travel through single file, one at a time. And that takes time. And so proteins, and with facilitated diffusion, what's going to happen is that these channel proteins can become saturated. There's a limited amount of substrate we can move in a certain amount of time, unlike passive diffusion where things can just enter at any point through the membrane as they please with no time or space constraints. That's looking at facilitated diffusion. And then looking at mechanisms that do require ATP, this is going to be our active transport. Or before we look at that, we are going to look at some of the different channel proteins we can work with. We have pores, which are always open. They're just little channels you can slip through, but like I said, like this would be a star pore up here, pretty much, because it's always open. The stars can come and go as they please, as the concentration gradient dictates. Then we also have mechanically gated. And those channels will open and close depending on like mechanical forces in the cell, like what is the size of the membrane? Is the membrane being pulled tight, or is it loose and swollen? There's also chemically gated pores, also called also called ligand-controlled gates, and these are how like neurotransmitters are received, pretty much, is that these gates will open when the correct substrate or ligand binds to a receptor, and then it will open. So let's say we have just insulin in the cell that's going to attach to the glucose transporter, and then it will let glucose into the cell. If there's an insulin plugged in, that opens the gate. Then there's also voltage gates. And voltage-gated channels are dictated by the electrical environment in the cell. Like the inside of the cell is relatively negative from the potassium ions and the chloride ions, and so the cell will maintain that negativity through voltage-gated channels. And then when we need to transport an electrical impulse, then we can start moving things around to get those channels opened. And those are just some different kinds of channel proteins we use for facilitated diffusion. Now let's have a look at the systems that do require ATP. This is going to be our active transport. And the defining feature of active transport is that it goes against the concentration gradient. Can't spell, that's okay. And so at least one thing is going to be moving against its concentration gradient. And so one example when we'd like to do this is kind of like what I was saying earlier with the sodium-potassium pump and wanting to establish an electrical gradient across cells. In order to reset back to zero, we have to flush out all of that sodium and we have to bring back in our potassium. And it requires ATP to do that, just because we're reestablishing a different gradient than what currently exists in the cell. And that is primary active transport, because it directly uses ATP in order to move things around. And if we have primary, we also have secondary. And so secondary is a little different in that it does not use ATP for itself. Instead, what it does is it uses another thing's concentration gradient in order to move something against a different gradient. So, like, we're trying to get something into the cell, but we also have something that's naturally trying to leave. So if we can get the thing that needs to go in into the channel, we can bring in something that needs to go out and use that gradient to change the shape of the channel in order to get the other thing to go in. That is called an antiport system, where one thing comes in and one thing comes out. It doesn't really matter which one is going against the gradient, but one thing in, one thing out is antiporting. And we also have symporting, which is the opposite of antiporting, which is two things in and at least one against the gradient, or two things out. We're just moving these things in the same direction, and one of them is going with their gradient and the other one is going against. And that is secondary active transport. All right, and so for the last part of this question, we are looking at osmolarity. And so what osmosis is osmosis is simply the diffusion of water. It's just the process of diffusion, and it's specific to water molecules because water behaves differently than anything else. And one of the defining characteristics of water in a biological environment is that water will follow solutes. So, explain the process of osmosis. So, osmolarity is different from molarity, not only because it has two additional letters in part of the word, but molarity looks at pretty much the number of moles per liter of solution. And what osmolarity is the number of particles per liter of solution. And this might not seem like a big difference, but let's say we have NaCl, both of which is sodium and chloride, both of which are very important ions for cellular processes. And so if we're looking at molarity, these are going to split up into sodium and chloride ions. And so we technically have 2 moles of solutes if we have 1 mole of NaCl in solution. But if we're looking at osmolarity, 1 mole of NaCl is just 1 mole, 1 osmole of stuff in solution. And so it makes our math a lot easier that way. And so the typical cell has an osmolarity of 300 milliosmoles. And so that's going to be kind of like our reference point. And so there's a variety of different solutions that we can introduce to a cell. The 2 main ones being normal saline, or NS, which is 0.9 NaCl, and D5, which is a 5% dextrose, or just essentially just glucose solution. And so both of these are also 300 milliosmol. And why that's important is because osmolarity is looking at the concentration of solutes in one cell versus the solution, versus just a solution, not necessarily together. So we have our NS, or D5, that is also going to be 300 milliosmoles. And because these are the same concentrations, these are referred to as isoosmotic. Iso meaning the same, osmotic referring to the osmolarity. And now there's also 0.45 NaCl solution. And so this solution would have 150 milliosmoles of particles. And so this would be referred to as hypoosmotic. Hypoosmotic, because it has less solutes compared to the cell. The cell is kind of like our reference point. And then we can also mix these 2 solutions together of the 0.9 NaCl and the 5% dextrose and that would have a total of 600 milliosmoles in solution, and that would be hyperosmotic because it has more solutes than the cell. So that's osmolarity, but we also need to talk about tonicity of a cell. And tonicity is looking at if we have our solution and we put a cell inside that solution, like we said earlier, that water follows solutes. And so if we put a cell in different concentrations of solutes, it's going to affect the water inside and outside the cell. So we have our NS solution. It's kind of our default. Say we have all these dots of solute, and the cell also has an equal amount of dots. And so this is what is referred to as an isotonic solution, because there's an equal amount of solutes both inside and outside the cell. Water is just going to move freely, and there's going to be no overall net movement of water, and so the cellular conditions and the outside are going to pretty much stay the same. And now you would think, putting our cell now in a solution of dextrose, is that we have all these bits of dextrose, but dextrose is a bit of an anomaly when we're looking at fluids, because we start with a concentration of 300 milliosmol outside and 300 milliosmol inside. But the thing about glucose is that each of these red dots is a glucose molecule. It's going to come inside the cell because glucose can do that, and Na and Cl cannot. They are referred to as non-permeable, while glucose is permeable. And so that's going to come into the cell and almost immediately be metabolized, and water is going to follow it in. And so that glucose is immediately metabolized and is no longer a part of our solution. But the concentration of glucose outside the solution still goes down. And so eventually all of this glucose is going to come inside the cell and then immediately be removed from our math, pretty much. So all of that glucose has gone inside the cell, and all of the water followed it as well. And so this is what is a hypotonic solution, because even though the glucose was isoosmotic compared to the cell, because of the fact that it moved water inside the cell without maintaining a stable equilibrium, pretty much, it was referred to as hypotonic. And so it just behaves a little differently. And then hypertonic were to be, let's say we have a cell in a solution of 2% sodium chloride, and so water would be drawn out of that cell and it would shrivel a bit, and that would be a hypertonic solution.